A good day, my name is EVALT LEBESE and I will be taking you through some of the work I did for my master's degree. The IPCC has been warming us of the Earth's surface and lower atmosphere, which have been warming up over the period or record approximately the past 150 years, and this warming has been attributed to an increase in the concentration of greenhouse gases as a result of human activities. The concentration of atmospheric carbon dioxide and other greenhouse gases have been predicted to continue in this century, reaching 600 parts per million or more, and the global average air temperature has also been predicted to rise by 2 to 5 degrees Celsius above the pre-industrial mean. These rising levels have been shown to influence the development and interaction of biotic organisms in many ways, including those with C3 and C4 photosynthetic pathway. Hence, the ability to predict how plants will respond to rising levels will help to assess the impact of global change on plant productivity and the competitive interactions. Recently, it has been noted that the tree Leucocidia sericea is encroaching into formerly treeless high-altitude C4-dominated grassland of the eastern escarpment in the Drakensberg mountains of KwaZulu-Natal. The expansion of high altitude of patches of the C3 grass Vestigia costata has also been observed. Now, the upward movement of these plants has been suggested to be due to rising temperatures. However, they could equally be due to elevated carbon dioxide, rising temperatures, both, or an interactive effect between the two. Thus, the aim of this study was set out to test the effect of elevated carbon dioxide and rising temperatures on the 6-month growth rate of Leucocidia sericea, triandra, and Vestigia costata. This was achieved by testing the interactive effect of rising temperatures and elevated carbon dioxide on the competitive interaction of the three species in the presence of other individuals of their own and the other species by monitoring their growth biomass and above ground growth rate. Leucocidia sericea is the only species in the genus Leucocidia in the family Rosaceae. This family is typically the family which provides the highest altitude trees in inframontane ecosystems. Leucocidia sericea usually grows as a small and evergreen shrub or tree. It grows in a broad range of habitat. As a result, it has a very wide distribution in high altitude areas of southern Africa. Vestigia costata also occurs in montane grassland in South Africa. The fibrous steels of this C3 grass species are considered by livestock farmers to be poor forage and unpalatable to livestock. However, it has substantial economic value to the local populations in southern Africa as it is used to make brooms and traditional baskets. Generally, very little is known about Vestigia costata. And triandra, which is commonly known as row-oat grass, is widely distributed and also occurs in a wide range of habitats, including areas with a wide range of rainfalls, altitude, and different soil types. It is also very well adapted to fire, uninterrupted selective grazing regimes, and has substantial economic and ecological value as it is also considered a keystone species. Leucocidia seedlings were sourced from Pietermaritzburg National Botanical Gardens at KwaZulu-Natal, and culms of both triandra and Vestigia costata grasses were sourced from Cathedral Peak in the Drakensberg mountain of KwaZulu-Natal in South Africa. This study, which is based on a factorial design of an interactive effect of elevated carbon dioxide and rising temperatures, was conducted at the University of the Witwatersrand in the Conviron walk-in growth chambers. Now, this table on your slide just sums up the complete experimental design with 3 replicates for each subtreatment in each chamber with different conditions. The 4 working chambers were randomly selected and used to allow for a complete factorial interactive design of 2 levels of carbon dioxide by 2 levels of temperatures. The two carbon dioxide levels were 400 parts per million, which is the current ambient level of carbon dioxide, and 650 parts per million, which is a level that could be reached later in this century. With regards to the temperature, a diurnal cycle of temperature was used based on ambient summertime temperatures at Cathedral Peak, where upward movement of Leucocidia and Festuca has been observed. The elevated temperature treatments were obtained by adding 2 degrees Celsius to this range, which is approximately the increase that is anticipated between the current temperature and temperatures corresponding with a concentration of carbon dioxide at 650 parts per million. The Leucocidia sericea seedlings and grasses of the triandra and costata were allowed to grow for 6 months, which is about the length of a growing season at an altitude of 200 meters on the eastern escarpment. The soil in the pots was a moderately fertile sandy clay loam consisting of 75% sandy loam and 25% potty mix, which is similar to the soil at the site where the plant material was sourced. The water regime was low limitation, administered every second day in equal doses, and the plants were grown in pairs in rectangular plant pots, which provided enough space to allow for an interactive competition with their own species and with the other species. Before and during the experiment, I measured the ground level stem diameter of Leucocidia sericea saplings, including the length of their stems, and also counted their number of leaves. And for each grass species, I counted the total number of tillers and also measured their height and their basal circumference. And at the end of the experiment, all the plants were harvested and the soil was gently washed off. Thereafter, I then separated them into roots, stems, and leaves, with each species separately, as well as the live and dead species separately. They were then oven dried and weighed to get their total dry biomass. The growth of Leucocidia sericea shrubs increased over time under elevated carbon dioxide, rising temperatures, and interaction between the two. However, between the treatments, only the effect of elevated carbon dioxide and rising temperatures alone enhanced the height of Leucocidia sericea over a 6 month period. The interaction between elevated carbon dioxide and rising temperatures did not enhance their growth rate, suggesting that the effect of either elevated carbon dioxide or rising temperatures are apparent when one is at an ambient level and the other at an elevated level. Similar response on the effect of elevated carbon dioxide and rising temperatures were also observed on the growth of Festuca costata, except that this was indicated by an increase in the number of tillers, which is associated with their growth rate. The response of Festuca costata to elevated carbon dioxide was expected, as C3 plants are favored under elevated carbon dioxide. However, their productivity is predicted to be reduced under rising temperatures, but I found that the growth of Festuca costata was in fact enhanced by rising temperatures. However, the accumulative effect of elevated carbon dioxide and rising temperatures on the total dry biomass of Leucocidia sericea and Festuca costata did not correlate with their growth rate response. For instance, the total dry biomass of both Leucocidia sericea and Festuca costata was disadvantaged by the rising temperatures and the interaction between rising temperatures and elevated carbon dioxide, which also did not correlate with their growth response. Hence, this suggests that elevated carbon dioxide alone has no effect on their total biomass. However, under the interaction of elevated carbon dioxide and rising temperatures, carbon dioxide seems to alleviate the adverse effects of rising temperatures on the total biomass of Leucocidia sericea and Festuca costata. In contrast to both Leucocidia sericea and Festuca costata, the growth response of the triandra grasses surprisingly correlated with the total dry biomass, in that they are both not enhanced by either elevated carbon dioxide, rising temperature, or an interaction between the two. and the response of Thamera triandra grossis to elevated carbon dioxide correlates with other studies' predicted response of C4 plants to elevated carbon dioxide due to the ability of C4 plants to assimilate up to 2,000 parts per million of carbon dioxide in their mesophyll tissues. However, the productivity of C4 plants is also expected to be advantaged by rising temperatures, as C4 plants are highly adapted to hot and high light conditions. Thamera triandra grossis that were grown under rising temperatures had all died on the third month of the 6-month growth rate experiment. Other studies suggest that low productivity and the reduction in the growth of C4 plants under warm temperatures is partly due to changes in their water relation that is caused by stress or low water regimes that is experienced by the plant. However, this was not a limiting factor on the response of Thamera triandra to rising temperatures, as the water that was supplied to all the plant in this experiment mimicked the rainfall water regime at Cathedral Peak, where tissues of Thamera triandra and Festuca costata grossis were collected. Therefore, the resulting high mortality of Thamera triandra under elevated or rising temperatures is independent of the effect of rising temperatures on Thamera triandra and the grossis of Festuca costata. They could have died possibly because they were transplanted at the end of their growing season at the beginning of this experiment. Lastly, when I looked at the inter- and intraspecific competition between the three species, I found that Leucocidia sericea saplings have a competitive advantage over both Thamera triandra and Festuca costata in terms of biomass accumulation, both under ambient conditions and in response to elevated carbon dioxide and rising temperatures. Additionally, there was no significant difference in interspecific competition between Thamera triandra and Festuca costata across any treatment, suggesting that Thamera triandra and Festuca costata can coexist regardless of changes in levels of carbon dioxide and temperatures. In conclusion, the growth rate of Leucocidia sericea and Festuca costata is apparently enhanced by small effects due to elevated carbon dioxide, rising temperatures, and not the interaction between the two. Their biomass was significantly reduced by the effect of rising temperatures, which are alleviated by the effect of elevated carbon dioxide. The growth of Thamera triandra was not affected by either carbon dioxide, rising temperatures, or the interaction between the two, suggesting that the adverse effects of climate change may not have detrimental effect on the growth rate and biomass of Leucocidia sericea, Festuca costata, and Thamera triandra. Rather, the effect of elevated carbon dioxide reduced the adverse effect of rising temperature on plants that are sensitive to rising temperatures. Therefore, these plants have the potential of occupying new areas. Thank you so much for listening to my talk.